everybody, welcome to your cell membrane and cell transport notes. So before we get into the membrane specifically, let's do a quick refresher on different types of cells. So we've already discussed prokaryotic cells, specifically bacterial cells, in detail. Now we're going to move on to eukaryotic cells. So these cells are much larger, they're much more complex, and they have lots of membrane-bound organelles. And some examples are plant cells, animal cells, and fungi cells. So here's a quick refresher on your prokaryotic cell. This should be a review for you from the previous unit. And now we're going to focus on these more complex plant and animal cells. So this hopefully is not brand new to you. Hopefully you did have some exposure to this in middle school, and you'll see a plethora of different organelles labeled there on this diagram. Things like mitochondria, chloroplast, Golgi or Golgi body, endoplasmic reticulum, which we short to ER, and then you see smooth and rough if you remember that, and a whole bunch of other ones. So we'll spend some time in class going over these organelles with an activity, but you should be aware of what these organelles look like and what their function is for the cell. The last one, though, that we will right now go over in more detail is the cell membrane. So the cell membrane itself is known by several other names, and you do need to know what all of these are. So it may be referred to as the plasma membrane, the phospholipid bilayer, or the fluid mosaic model. And the membrane itself is selectively permeable. So this means it's choosy. It doesn't just let everything come in and out of the membrane. So it allows things like nutrients into the cell, and it also allows for waste to move out of the cell. So let's break down the name phospholipid bilayer a little bit more. So in terms of the prefix phospho, this is referring to the phosphate head. And the head is hydrophilic. So by hydrophilic, that means water, hydro is water, and philic is loving. Okay, so the phosphate heads like to be where there's water, which is a very large percentage of the body. Lipids make up the tails, and these are hydro, once again, water, phobic. So this time the tails are water fearing. So they actually align themselves together. So it's also a bilayer, meaning it's two layers sandwiched. So if you imagine my fingers are the tails face each other, and the heads are on the outside where my wrist would be. And this is how the cell membrane is formed with those two layers. The other term we talk about in terms of the cell membrane is the fluid mosaic model. So when you hear the word mosaic, you probably think about something like that, maybe some stained glass or some sort of beautiful structure or picture of a bunch of different pieces assembled together. Well, in the cell membrane, there are many different molecules that are floating throughout this membrane, which you see in this picture here. So the membrane is all the yellow, that's the heads, and then the tails are in the middle. But then you see all these different blue structures and green and purple. So those are those molecules. And what are they? They're things like proteins, cholesterol, and carbohydrates. All right, now we're going to get into the details. So in terms of the proteins that can be in a cell membrane, there are 5 that we are going to focus on. So you'll see them all listed there on the left. You have the glycoprotein, the integral protein, peripheral protein, channel protein, and carrier protein. So glycoprotein, those ones have carbohydrates. So glyco is a prefix meaning carbohydrate, so it's attached to them like little antenna. An integral protein is within the whole membrane, so top to bottom, it goes through the whole thing. It's like integrated, right? A peripheral protein is on the outside edges or borders of the membrane. So think about your peripheral vision, like what you can see out of the corner of your eye. Peripheral proteins, same concept. They're on those outside edges. The channel protein is just that. It's a channel. Imagine like a boat channel. It allows certain things to flow through. So it's same thing. And then a carrier protein, these ones are the most fun to me because they look like little Pac-Men. So they'll be open towards the top, They grab something and then they open on the bottom to release it into the cell, or it works in reverse out of the cell. So here is your picture, and take a second and pause it and then try to identify all 5 of those proteins in this diagram. All right, so now that we know what they are, we've talked about where they're located. Now let's talk about their functions. So glycoproteins and peripheral proteins help transmit signals, okay? The structure of the cell membrane is supported by integral and peripheral proteins. And then you have your transport proteins, which go with cell transport, which we'll get to in just a minute. Those ones help move things in and out of the cell. Cholesterol is another important component, and this is found in between the tails. So it prevents those tails from sticking together, but it also helps provide a little support, which is more rigid than the typical lipids in the membrane. And it can be up to 50% of your entire cell membrane. Carbohydrates then are the final thing, and these help identify those signals. So carbohydrates can be attached in one of 2 ways. They can be attached to a protein, so then they're called a glycoprotein, or they can be attached to one of the heads of the actual cell membrane, in which case they're called a glycolipid. But either way, think about signaling like an antenna on a car or something like that. It's what they do is they help with signals. So now that we've discussed the cell membrane in detail, let's get into the transport of things through that membrane. So there are 2 major types of transport that can occur: passive transport and active transport. So you know what active means, right? So passive is essentially the opposite of active. There are 3 types of each, which we will get into in detail. So passive transport, this is movement from high to low or with the concentration gradient. So we tend to reference a hill a lot. So if you were running and you come across a hill, which time are you going to use more energy? Or actually, let's say biking, just because you don't even have to pedal to go downhill, right? So if you're on your little bicycle at the top of the hill here, excuse my terrible drawing, and you're going downhill, you don't need to use any energy to go downhill. So you're going with the concentration gradient, and you're working towards equilibrium, or that flat ground, and it requires no energy. So the examples of passive transport are dependent on what is moving through the membrane. So diffusion is focusing on lipids and gases in particular. Facilitated diffusion can be many particles. It's usually larger particles that can't just slip through the head, so like larger food particles and things like that. They move across using channel proteins, but with no energy. They're moving from high to low concentration. And then lastly, osmosis, which is the movement of water. And the easiest way to remember osmosis goes to water is H2O is water. And oh, look, osmosis starts with O. So diffusion rates, remember, diffusion are lipids and your gases, can be affected by several things that you need to be aware of. Things like temperature. So higher the temperature, the faster the diffusion rate. Pressure, particle size, collision number, and the diffusion medium. So if it's in a solid, it's going to be a lot slower of a diffusion rate than if it's in a gas state. All right, let's practice. So pause here and label these 3 types, and then we will check your work. All right, let's see how you did. So number one is facilitated diffusion. So if you notice that high to low concentration, there's many more of these little orange cubes outside of the cell than inside. So by moving to the inside, you're moving with the concentration gradient. Number 2 is diffusion. So you're seeing CO2 and O2, which carbon dioxide, oxygen, those are both gases, so they're moving through. And then last but not least, number 3 is osmosis. This is the movement of water. It's talking about red blood cells, and when they're placed in water, how the water moves depending on whether it's a distilled water concentration solution or a salt water concentration. A little bit more too about osmosis in cells is the words hypotonic, isotonic, and hypertonic. So when you see hypo, I want you to think hippo, right? Hippos are big and they're round and they're swollen. So this is when water moves into the cells and the cells swell, like you see, like you see here and here. Now, plant cells like this because they have a cell wall to keep their structure. Blood cells, on the other hand, don't like this. They can actually burst. Isotonic, then, is water moving in and out at the same rate. People mistakenly think that isotonic means no movement at all, and that's not true. It's moving in and out just at the same rate. And then lastly, hypertonic. So think about maybe someone's like that's really hyper, they're always on the go, they're always moving. They're probably pretty skinny because they're always like moving around and running around and acting crazy. So hypertonic is when water moves out and the cells as a result get shriveled. So now that we've talked about passive, let's talk about active. And we're going to go back to our little bicycle here. So this time you're at the bottom of the hill and you're like, oh man, I'm so tired. I don't want to ride anymore. I just want to go home. But guess what? You got to go up that hill to go home, and it requires energy to do so. So active transport is the movement from low to high or against the concentration gradient. And there are 3 major types and then a 4th one I'm just going to mention so you're aware of. So transport proteins, with those transport proteins, the only difference between active and passive of transport proteins is what your concentration gradient is. Is it against it or with it? So in this case, it's against it. Then you have endocytosis, the prefix endo meaning in. And this is where the cell engulfs something. So there's 2 types. There's phagocytosis, which is eating, and penocytosis, which is drinking. Then you have exo, so exit or out. Exocytosis is leaving. So this is when material is moved out, and this is really common for cells to remove their waste. And then the last one I want you to know about is just receptor-mediated endocytosis. So this is super specific. It binds to a specific protein that it's looking for, and then it uses endocytosis, the process of engulfing, to bring it into the cell. All right, time to pause again. Let's practice. Take a minute and identify these 3 types. All right, let's see how you did. So, number 1, you should have gotten exocytosis. Those particles are leaving the cell. Number 2, we see those little purple Pac-Men, right? That's transport proteins. And then number 3 is endocytosis. That's your engulfing. So one final thing I want to touch on is cell metabolism. So metabolism itself is the process by which organisms like us, we use food, water, nutrients to make energy. So cells do this in 2 ways, through anabolism and catabolism, okay? So in anabolism, they build macromolecules from simple molecules, right? Complex macromolecules, proteins, carbs, etc. They're building that, and this uses energy. So an example of this is photosynthesis. This process creates and then ultimately stores glucose for the plants. Catabolism then is the opposite. This is the breakdown of macromolecules into their simple compounds or monomers. Throwing that word out there from the earlier in the year, right? And this is how they release energy. So cell respiration is an example of this because glucose is being broken down to provide energy for the organism. So just touching on that now, we'll get into that in much more detail because next up we'll be jumping into photosynthesis and cell respiration in more detail. So that is it for your